Hi, today we are going to discuss the gluconeogenesis. Before to discuss the gluconeogenesis, there are three important concepts related to the gluconeogenesis. These are, number one is a glycogenesis. The second one is a glycogenolysis. The third one is a glycolysis. And the fourth one is that we are going to discuss gluconeogenesis. What is mean by glycogenesis? After the meal, blood glucose level become higher than the normal range. When the blood glucose level become higher, then this blood containing high amount of glucose when reach to the pancreas, beta cells in the islet of Langerhans release special hormones called insulin, and the insulin is the hormone which convert glucose into glycogen and normalize the blood glucose level. This is the function of what? This is the function of insulin. Remember, glycogen is a polysaccharide, and the glucose is stored in the body of animals when it is present in excess amount in the form of glycogen. Now, when we have less amount of glucose in the blood, when the blood glucose level fall, then what happen? Then the glycogen is converted back into glucose. But how? When this blood containing less amount of glucose reach to the pancreas, alpha cells in the islet of Langerhans of the pancreas release hormone known as a Glucagon. What is the function of glucagon? Glucagon convert or break down the glycogen back into glucose to normalize the blood glucose level. Remember, when the blood glucose level is higher, this condition is known as hyperglycemia. And when the blood glucose level is low, then the normal range, then it is known as a hypoglycemia. So these are opposite of each other. In the glycogenesis, what happened? Glucose is converted into glycogen. In the glycogenolysis, what happened? The glycogen is converted back into the glucose to normalize the blood glucose level. And the glycolysis, what happened? What is glycolysis? When a body have less amount of ATP or energy, or when a body have high amount of AMP, or when a body contain high amount of glucose in blood, when blood glucose level is higher, then glycolysis occur. So insulin promote glycolysis. In glycolysis, the glucose molecule is broken down into pyruvate, and less amount of energy is released in the form of ATP. So glycolysis occur for different purposes. The one of them is to release energy, and also by the glycolysis the blood glucose level become lower or normalized. The third term is gluconeogenesis. What is mean by gluconeogenesis? Actually, gluco mean glucose and new mean new. substances. And genesis means synthesis or generation. So the synthesis of glucose from new substances, from new precursors, is known as gluconeogenesis. What are these new precursors? These precursors are, it may be pyruvate. Pyruvate can also bake to convert into glucose. Protein. Protein can also be converted into glucose. Lipids. These may be lipids, lactic acids, alcohol, or other intermediate of the Krebs cycle. So these can be converted into glucose when the blood glucose level is lower. When gluconeogenesis occurs, when the blood glucose level is lower than the normal, then gluconeogenesis occurs to synthesize or prepare glucose to normalize the blood glucose level. Now we are going to discuss the gluconeogenesis and glycolysis in full detail. Remember one point here. Insulin hormone favor glycolysis and glycogenesis. By the glycogenesis, the glucose level become normal, which is higher in the case of hyperglycemia. And by the glycolysis, the glucose is broken down into pyruvate. So the glucose level, if it is higher, then it is released by the glycolysis. So both of the processes are promoted by the insulin. In the glycogenolysis, the glycogen is converted into glucose, so glucose is formed, and it is same as in the gluconeogenesis. But in the gluconeogenesis, the substances which are used to form glucose are not carbohydrate. These are not carbohydrate or glucose substances. These are other substances, for example, protein, lipid, lactic acid, alcohol, and so on. And in glycogenolysis, the substances are carbohydrate. Glycogen is carbohydrate. This is the difference between both of them. Now we are going to compare glycolysis and gluconeogenesis. This is glycolysis, and on this side this is gluconeogenesis. As we studied in glycolysis, glucose is broken down into pyruvate molecules to release less amount of energy. Glucose is broken down into pyruvate molecules to release energy in the form of ATP. In the glycolysis, glucose is broken down into pyruvate molecules. If the glycolysis is aerobic, then the pyruvate goes under other reactions such that link reaction, Krebs cycle, and electron transport chain. And if it is anaerobic, then the pyruvate is converted into lactic acid or alcohol. Now, when glycolysis occurs, when the condition is hyperglycemia, when there is high level of glucose in the blood, or we need ATP, then glycolysis occurs. Actually, glycolysis is completed in 10 steps, in which total 10 enzymes work. All of the steps of the glycolysis are the reversible of gluconeogenesis, but only 3 steps of the gluconeogenesis are different from the glycolysis. These are not reversible steps. These are quite different and using different enzymes. In gluconeogenesis, the glucose formation or glucose synthesis occur. Glucose are synthesized from new substances which are not carbohydrates, which are not glucose. These substances may be protein, lactic acid, or lipids, or alcohol, or other intermediate of the Krebs cycle. So 3 steps, step number 1, step number 3, and step number 10 of the gluconeogenesis are quietly different from the glycolysis. All the other remaining steps are actually the opposite or reversible step of the glycolysis. Now we are going to convert the lactic acid into glucose, which is not carbohydrate source, then protein, lipid, pyruvate. As we know that lactic acid is formed in the muscles, and this is carried into the liver or the cortex of the kidney. This lactic acid is converted into pyruvate molecule in the presence of lactate dehydrogenase enzyme, which remove the hydrogen from the lactate and add into the NAD to become NADH. So here the oxidation of lactic acid occurs. Lactic acid is converted into pyruvate molecule. Then the pyruvate molecule is converted into oxaloacetate or oxaloacetic acid. Oxaloacetic acid contains 4 carbon and pyruvate molecule contains 3 carbon. So where from this carbon came? This carbon came actually from carbon dioxide. Carbon dioxide, ATP and vitamin biotin are used in this step. And this step is catalyzed by an enzyme known as pyruvate kinase or PC. ATP is converted into ADP and inorganic phosphate. Now the oxaloacetate, this is very interesting point. The oxaloacetate cannot leave the mitochondria because the membrane of the mitochondria is not permeable to oxaloacetate. Therefore, oxaloacetate must be converted into other substance or molecule to leave the mitochondria, to reach the cytosol of the cell and convert into the glucose. Therefore, oxaloacetate convert into malate. Malate, the membrane of the mitochondria is permeable to malate. So malate leaves the mitochondria and come out into the cytosol where it converted back into the oxaloacetic acid. Now the oxaloacetic acid is converted into PIP, phosphoenol pyruvate, which convert into pyruvate. The phosphoenol pyruvate contains 3 carbon while the oxaloacetate contains 4 carbon. 1 carbon is removed in the form of carbon dioxide. In this step, GTP is converted into GDP. So this phosphate from the GTP is added into the phosphoenol pyruvate. And this step is carried out by an enzyme known as PEPCK, phosphoenol pyruvate carboxylase kinase. Now this PEP is converted into 2-PGA, 2-phosphoglyceraldehyde or 2-phosphoglycerate, which is converted back into the 3-phosphoglyceraldehyde. It is converted back into the 1,3-bis-phosphoglyceraldehyde, which is converted into G3P into fructose 1,6-bisphosphate, and there this step is also not reversible. So here different enzyme fructose bisphosphatase is used to convert fructose 1,6-bisphosphate into the fructose 6-phosphate, and then the enzyme phosphoglucoisomerase convert fructose 6-phosphate into glucose 6-phosphate, and glucose 6-phosphate cannot be converted into glucose by hexokinase. Therefore, other enzyme is used known as glucose 6-phosphatase. But this enzyme is present inside the smooth endoplasmic reticulum. So the glucose 6-phosphate move into inside the endoplasmic reticulum, where the glucose 6-phosphate convert it into glucose, and it is released into the bloodstream to normalize the blood glucose level. So this is the conversion of lactic acid into the glucose. Now the precursor may be lipid. If it is lipid, we know that there are different types of lipid, but here we are focusing on the triglyceride. Triglyceride is actually a simple lipid. It contains glycerol and 3 fatty acids. This glycerol is phosphorylized into G3P, and G3P is converted into fructose 6-phosphate, then fructose 6-phosphate, then glucose 6-phosphate, then glucose, and move to the blood. But here this is important. When the glycerol is converted into G3P, it can move in this direction to go under glycolysis, or it can also move in this direction to go in and under the gluconeogenesis. This is very important. The fatty acid of the triglyceride is converted into, if it is odd chain fatty acid, then it is converted to succinyl coenzyme A, which is the intermediate of the Krebs cycle, then into succinate, it is also intermediate of the Krebs cycle, then fumarate, it is also the intermediate of the Krebs cycle, and then finally into malate, this is also the intermediate of the Krebs cycle. It leaves the mitochondria, comes out into the cytoplasm, then converts back into the oxaloacetate, which convert into PEP, and PEP into 2-PGA, 3-PGA, and then to 1,3-dive-PGA, then into G3P, then fructose 1,6-diphosphate, fructose 6-phosphate, glucose 6-phosphate, finally to glucose, which move into blood. If the source is protein, we know protein have amino acid. These are the building blocks of the protein. So these amino acids, if these are glucogenic amino acid, if the amino acid of the protein are glucogenic amino acid, then these convert into alpha keto acids or other intermediate of the Krebs cycle, and alpha keto acid converts into pyruvate into oxaloacetic acid, which convert into malate leave the mitochondria, converts back into oxaloacetic acid, which convert into PEP converts into 2-PGA, into 3-PGA, into 1,3-di-PGA, into G3P, into fructose 1,6-phosphate, into fructose 6-phosphate, into glucose 6-phosphate, finally to glucose, and reach to the bloodstream to normalize the blood glucose level. So actually gluconeogenesis occurs to form glucose from non-carbohydrate substances. As I discussed, these substances may be protein, lipid, lactic acid, alcohol, or intermediate of the Krebs cycle. So this is something about the gluconeogenesis. If you like, please subscribe to our channel and share with friends. Thank you so much.